everybody, my name is Julia Diamorum. I'm a PhD candidate in the biochemistry program at Emory University. I work in Dr. Anita Corbett's lab. Our lab uses various methods and model systems to investigate protein-protein interactions in relating to a neurodegeneration.
I'll show you around the lab. And I'm going to also show you where we keep our neuronal cells. I keep the cells in an isolated room in its own incubator. This is the hood where we do our experiments. This is the confocal microscope. The cells thrive best at 37 degrees Celsius or 98.6 degrees Fahrenheit, otherwise known as our body temperature. I use the confocal microscope daily to check the health of growing neurons. This image shows a healthy population of neurons. In gene expression, your permanent genetic code, your DNA, is transcribed or copied into RNA. RNA can perform many functions, and this species of RNA, called mRNA, is being translated into proteins. In many genetic diseases, a mutation in DNA can lead to a change in the corresponding protein. Although this change may seem minor, it can alter how the protein works and trigger many downstream effects. For example, changing one amino acid, or a single building block of a protein, can be linked to neurodevelopmental and neurodegenerative diseases. Biochemical evidence shows that this substitution can interfere with protein-protein communication. During normal function, a protein will interact and communicate with other proteins. When there are genetic alterations to a protein, the interactions will be perturbed. I use biochemistry to study the way these molecules communicate. After I collect my neuronal cells, I will break them open and isolate the proteins within them. I use antibodies to target a specific protein. The antibodies are conjugated to magnetic beads. I can isolate the beads, antibodies, and target protein with a magnet. All the proteins communicating with my target protein will be in the sample too. Here's a common way to visualize these interactions. Before the immunoprecipitation, I collected my cells and checked if the interacting proteins were expressed in the neurons. I used a negative control, 2 positive controls, and a sample of the neurons that contained the genetic mutation. I labeled it diseased. This visualization method is called a Western blot. The thick black band shows me that the protein is present. In the diseased neurons, the interacting protein is present. What happens when I use antibodies and magnets to pull the protein out of the neurons? Again, I included a negative control, 2 positive controls, and the mutant neurons in the immunoprecipitation experiment. If the experiment worked properly, I should see no interaction in the negative controls, and I will see interaction in the 2 positive controls. At this point, I can only predict what the diseased sample will look like. Indeed, the proteins do not interact well in the diseased neurons. The band is not thick and dark like it is in the positive controls. Biochemistry helps scientists understand underlying molecular mechanisms in diseases, including neurodevelopmental and neurodegenerative diseases. The more we understand, the better we can engineer drugs and other therapies to help patients. Thank you for your attention. I hope I convinced you how cool and important biochemistry is to scientific research. Enjoy the brain bee.